is episode 182 of the Stem Cell podcast, Stem Cells and Society, with Dr. Christopher Scott. Hey everyone, we are Daylon and Arun. Welcome back to the Stem Cell podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. We try to pick top stem cell researchers to interview on the podcast, but we want to know who you want to hear. If you know anyone that would make a great guest, send us your suggestions at info@stemcellpodcast.com. Today, we have Dr. Christopher Scott from Baylor College of Medicine. He's on the podcast to talk about his research on the ethical, legal, and social implications of emerging biotechnologies. We've also got our usual roundup of recent highlights in stem cell news coming right up.
But first, at the beginning of 2020, which, can you believe it? That was like 10 months ago, not 10 years ago. Stem Cell Technologies conducted a survey asking scientists to help highlight the needs and challenges in the human pluripotent stem cell field. You can read the survey results to learn more about the most interesting insights on topics such as reproducibility, quality control, and how to address them in your own research. So you can visit www.stemcell.com/HPSC survey results to learn more. And speaking of stem cells and society, we're actually going to do something a little different with the first segment here today. This is a unique episode. We're talking about stem cells and society and bioethics of stem cell biology and other technologies. But I would say when it comes to policy news, there's a huge piece of policy news that has recently emerged from here in California, and you've probably heard about it if you're in the stem cell field, and in particular if you're here in California like me. Proposition 14 has passed here in the state, in the Golden State. And what that means is that the California Institute for Regenerative Medicine is coming back. It's going to receive another $5.5 billion of funding over the next 10 to 15-ish years or so. It was narrowly approved by just a few 100,000 votes. It was, a part of the very recent November election that we had here and nationally. It's narrowly approved, 51% to 49%, but that's all you need. So the people have decided, the people have spoken, and they are going to infuse researchers and clinical trials with billions of more research funding and trial funding to actually study some areas that don't even involve stem cells. I mean, this is going to be, stem cell centric funding, because that's what STEM is all about. But a lot of this money is actually going to be pumped into studying neurodevelopmental disorders like Alzheimer's research, Parkinson's, and it's a huge boost for biomedical research here in the state. Proposition 71, which was passed back in 2004, it put California on the map for everything stem cell related. And part of the reason that Prop 71 was passed back in the day was because here in the U.S. there was a federal ban on embryonic stem cell research. So California took it upon themselves to be like, all right, we're gonna if you're gonna ban embryonic stem cell research on the federal level, we're going to take over here at the state level and make California the place to be for stem cell research. And it has been. I'm actually a beneficiary of CIRM. My training was heavily funded by CIRM. I would say I'm a stem cell biologist today because of the California Institute for Regenerative Medicine. So I'm obviously very grateful here. And now that Prop 71, which was again passed in 2004, has ran out of money. It ran out of money last year. We're very fortunate to have Prop 14 come in and fill the gap. So if this didn't pass, CIRM would go under. And for obvious reasons, for the stem cell community here in California, we are very happy that it did pass. And we're actually going to be having a CIRM guest here on the podcast in a few weeks, Dr. Kelly Shepard, who is a senior level employee at CIRM, more focused on the science of stem cell biology and better understanding why certain things need to be funded and why certain areas of stem cell research are priorities for CERM. So we'll get her perspective on things, and in particular on the passage of Prop 14. So yeah, I mean, it's just, something I wanted to drop here on the podcast before we dive into the Roundup papers. But it is a huge, huge deal for those of us here in the U.S. and those of us here in the Golden State of California.
Yeah, Arun, I was very pleased to hear about this because honestly, I feared the worst. I fear it would go the other way. And particularly because what you mentioned there, was born out of this really hostile environment toward embryonic stem cell research. And I thought that was the major impetus back then. I mean, I'm sure it was. That got it through. And I thought, absent that, and given the alternatives to embryonic stem cell research, that a lot of the populace there in California would vote a different way. But, I have a couple explanations that may account for it. I mean, whether they're right or wrong. I mean, I'm guessing that, one, it's just, you get in the habit of it, perhaps, or you see the good returns, and maybe you think, well, there's nothing wrong with this, having this, even absent this hostile environment, let's keep it going. I think that maybe was the mindset of a lot of the voters there, seeing how maybe there were some positive returns, both to the academic, prestige, also the retention of great minds, the recruitment, some returns in the economy, perhaps. But also, I wonder if it was the ability and willingness of the organization and the scientists there to kind of pivot, Initially, there was a focus on, whatever the focus was. I think we talked to one of the administrators on the podcast, and he was sharing with us how there was a bit of a pivot to focus on things that were more applied, things that would have a return in the shorter timescales and maybe be more visible to the voting public. And now you can see even that they're pivoting again to have a focus on these neurodevelopmental research areas. So I feel like this is another testament to California's just openness and willingness to be flexible in their approach, and I think it served them well.
Yeah, I think so too. I think it was somewhat of a perfect storm. And to be completely honest with you, I think most of us here in California were just shocked. We were shocked to see it pass. We were not expecting it to see it pass, especially now during the COVID era, considering that there's so many other things that are happening in the world. Stem cell biology tends to take a back seat, right? And it wasn't as popular as the initial Prop 14, the initial Prop 71, which I believe passed at 60% yes to 40% no, and now it's about 51% yes. But perhaps flying under the radar helped out. Honestly, if you think about it, and I was actually looking at the money, there weren't really any negative attack ads against Prop 14 this time, whereas last time there were quite a few. So perhaps flying under the radar worked out.
Yeah, well, one guy who's going to benefit from that is Calvin Kuo, who just had a big paper on lung organoids and COVID, but we're not going to talk about that. I can't talk about COVID anymore. So we're going to talk about something else that didn't come out of California. I'm going to start with something near and dear to my heart, which is a little convergence of the blood and the skin. I love the blood, and I have some skin. I have a lot of skin. So do we all. So I'm going to tell this story about epidermal stem and progenitor cells, okay? This is what gives rise to the stratified epithelium in embryonic development. And epidermis, which is made up of the epidermis and the mucosa, they make up barriers that are like, that's your first line, right? You have the keratinocytes, that's that tight connected layer, and there's a lot of diseases that have their origins in keratinocytes and other elements of the skin. So there's like carcinogenic exposure, of course, it hits the skin, right? You have UV light's the obvious one, but also chemicals, diseases like HPV. They're all risk factors for squamous cell carcinoma. But there's not really a good understanding of the kind of genetic or cellular underpinnings that underlie the disposition, a predisposition to squamous cell carcinoma, okay? Specifically in the case of Fanconi anemia, all right? Fanconi anemia, most people would recognize that as a disorder in the blood and the hematological system. It's loss of function there's a lot of different genes that give rise to Fanconi anemia, because it's all genes that are involved in DNA interstrand cross-link repair. So these cells are really susceptible to DNA damage. Oftentimes you'll see this results in hematopoietic stem progenitor cell death, so you get bone marrow failure, but also you'll get, progression to leukemia. You'll get these oncogenic transformations. But what no one talks about is these patients are also very highly predisposed to early onset of aggressive squamous cell carcinoma. Okay, so above 10% of these patients in this study, which was by Suzanne Wells out of Cincinnati Children's Hospital, out of 105 Fanconi anemia patients, 11.4% reported a history of skin cancer, okay? And that stands out because these patients aren't typically known for diseases outside of the hematopoietic compartment. And also, there's not the same degree of, like, in the hematopoietic system, you have the constant turnover, which can lead to exhaustion. Not so much the case in the epidermal stem cell compartment. All right, so this led Suzanne and her group to explore the Fanconi anemia epidermal specific pathologies, okay, because they hypothesized that they exist for two reasons. One is that when you do bone marrow transplant to treat Fanconi anemia related bone marrow failure, it doesn't mitigate the risk of squamous cell carcinoma. In fact, it increases it. And if you look at the Cancer Genome Atlas associations between certain gene mutations and squamous cell skin cancer, it's like 60% of the FANC genes are associated with skin cancer, but none of those, 1% of those are associated with leukemia. So there's a lot of reasons to think that there's these predisposition to squamous cell carcinoma in the case of these FANC patients. In order to explore that, what they did is they took patient-specific, FANC patient-specific iPS cells, directed differentiation into these epidermal stem progenitor cells, and then also differentiated those into epidermal organotypic rafts, okay? So there's these raft tissue models that were like organoids of the skin. And what they found is that while the FANC loss of functions didn't affect the growth or differentiation of those epidermal stem cells, once they were engineered into the rafts, you had they were all banged up. There was all this disruptions of tissue adhesion and increased proliferation of basal cells. And then when you looked into the human patients, they had this human cohort of 105 patients. When you looked into them, they saw the similar ultrastructural pathologies and other structural defects in the chromosomes, and non-chromosomes, desmosomes and hemidesmosomes. So they actually had it down to the organelles, the defects underlying the pathology there. So altogether, the data kind of pretty much once and for all shows that these FANC individuals, they're prone to the skin blistering, but it's because of the mechanical stress and DNA damage related to the FANC anemia and ability for tissue repair. And now, having created this kind of raft model, they have a nice means of looking downstream and seeing what the mechanisms of this are, and of course, at the end of the day, trying to find effective therapies to prevent and or reverse this susceptibility to squamous cell carcinoma, which ultimately leads to death in many cases. You wouldn't think it, but a lot of these FANC patients die from squamous cell carcinoma that could be avoided.
Yeah, connection between blood and skin here. It's another amazing paper coming out of Cincinnati Children's. If you haven't heard by now, their stem cell and organoid team over there is absolutely world class. So many incredible researchers there, like Takanori Takebe. They just hired Mingxia Gu on the vascular side of things, Suzanne Wells, James Wells. So They are the place to be for organoid biology. One thing to consider here, I really like these rafts. It's another really cool tissue engineering approach to study this particular disease. One thing that they did mention was that a limitation of the raft system is that you're only really studying keratinocyte biology. You're not focusing on any other cell types that may be critical or contributing to the disease pathology, like the immune cells or the stromal cells. But that was a big plus for me. I really like that system.
Yes, it's impressive. And I mean, of course, we're moving towards these, really heterogeneous collections of cells that are assembled into organoids. I feel like that's the present now. It's not even the future. But with something like Fanck, I think that the Fanconi anemia, I think the really important insight here is to really highlight and elucidate the cell intrinsic defect, because it really is a disease of the cell and the DNA repair process. And just to echo your praise of the people at Cincinnati Children's, I want to point you guys towards the episode we did with some of the trainees there. They invited me out to visit for one of their Graduate Research Council days as part of the podcast. I gave a great talk. Well, I gave a talk. I don't know if it was great, but it was a lot of fun. I had a great time there, and we interviewed the trainees. So check that out after you hear this, all right?
I'm sure it was a great talk, Daylon. Don't sell yourself short. Moving on to actually another cell stem cell paper, a brief report, Cell Surface Mechanics Gate Embryonic Stem Cell Differentiation. First author of this paper is Martin Bergert, and last author is Alba Diz Munoz. This is coming from the EMBL, the European Molecular Biology Laboratory, which are actually spread out, I think, across different places in the EU. But Dr. Diz Munoz, I believe, is in Heidelberg, Germany. So we're talking about differentiation here, right? We like talking about differentiation either for studying basic cell biology or for translational purposes, right? You can make all sorts of stuff from embryonic stem cells. But differentiation usually happens with shape transitions. If you think about it, a pluripotent stem cell looks different from a differentiated cell, right? And these shape transitions help enableize the specialized function of a cell. Okay, so like a cardiomyocyte, my favorite cell, is like a little brick-shaped cell, and it's brick-shaped for a reason. It helps for the contraction, helps with the force generation, and the stem cell itself, the ancestral stem cell, is different when it comes to shape. But to change shapes, cells have to change the mechanical properties of their surfaces. But whether cell surface mechanics can actually control the process of differentiation hasn't been super well explored. We have covered a few other papers here on the podcast that have talked about the important role of the cytoskeleton and cell contact with the matrix in influencing differentiation. For example, we actually covered, I believe, the Millman Labs paper recently. I forget where it came out, but they were able to show by modulating the cell cytoskeleton and the cell contact with the matrix, you can actually improve pancreatic beta cell differentiation and function. They can make more insulin. And this is more of a pure basic science study, but they are showing here that membrane mechanics can actually gate the exit from the naive pluripotent state of mouse embryonic stem cells. Naive pluripotency is a pretty hot topic these days, right? It's this powerful state of pluripotency that a lot of us are striving to get towards, because you can make perhaps more and better different types of cells from these naive state cells as opposed to primed, which is the alternative. So they can measure membrane tension during the early differentiation process using a pretty cool technique here. And they can find that the naive state cells actually can release their plasma membrane from the underlying actin cortex when actually transitioning to the primed state. And what they're able to do is you can actually mechanically tether, so-called tether, the plasma membrane to the cortex by enhancing the activity of a protein called esrin, or actually expressing a synthetic linker. And you can demonstrate that this, preventing this detachment is actually forcing the stem cells to keep their naive state. So you're talking about maintaining naive pluripotency, as I mentioned, is something to strive for. And ultimately, they're able to show that a decrease in this membrane to cortex attachment is a cell intrinsic mechanism that's really needed for the stem cells to exit pluripotency. So it's a pretty straightforward, relatively simple paper here. It's got two main figures, but it's characterizing these cells as they transition from the naive to the prime state. And it's really this the integrity of this membrane attachment to the matrix that's driving the transition between the two states. So I'm a fan of this area, mechanobiology. I think it's something that we really need to pay closer attention to when it comes to our cell culture. We grow our cells in artificial substrates, right? We use matrix, we use matrigel, we use gelatin, we use plates made of plastic, right? Which are not exactly the same thing as, what's happening in the real body. So these are important things to really think about. How is mechanobiology really influencing stem cell function? And I think it's a really quite a hot topic these days.
Very hot topic, my guy. Very hot topic. Although I must say it takes a lot of courage to present a paper on this show that doesn't have any organoids. There is no single cell. I mean, what is going on? How dare you?
Sorry.
It's good, though, because it's basic. It's conceptual advance. And for me, the conceptual advance here, it was a conceptual advance in the past, these early days. Studies showing that shape really mattered. I know Nina Bissell first showing that you had mammary gland epithelial cells and in monolayer was one thing, but you had to get them to form the shape of a duct. You had to get them in the right shape in order to make the casein, So function is form. And later on showing that differentiation of pluripotent stem cells or any kind of progenitors down different lineages was oftentimes dictated by, whatever, the elasticity of the matrix or the shear or whatever, these forces. And here, the conceptual advance to me is all that made sense, is it dictated where you were and it dictated you what you should be like. But here it's in the cell. Mm-hmm. It's, the shape you are. And it's something that it was shown scientifically then as soon as they give you the proof. You're like, oh, yeah, because like you said at the very beginning, the cells just look different. And some of the people who have the most amazing clinical expertise in the world, Shukret Metalipov, for one, a guy who's the only one who can do what he does. There's a guy in my center here who does ICSI, who kind of invented it, who he could tell you there's like 100 sperm in there and they're all bad. But they're not bad visibly, but he could tell you, he has the intuition to tell you which is a sperm to shoot in there. So on an intuitive level, on an instinctual level, as a scientist, what you see oftentimes is what you get, and there's some scientific proof for that.
Science is complicated, yet simple and beautiful, right? It makes sense. It just makes sense that if differentiation happens, it's influenced by, of course, these intrinsic variables like what we're talking about here, and also a variety of extrinsic variables. And our job and your job out there as trainees, as listeners of the podcast, is to figure out how all of these things work. And for those of us working in vitro biology, how can you best mimic all of these variables and incorporate all of these variables into your pseudo artificial in vitro culture? Good luck.
Good luck. Well, sometimes in vitro is just not the path, and this is a story that speaks to that. It's another blood story and the skin, the interface. I'm loving it today. It's about tissue-resident memory T cells. There's a ton of immune cells out there. These are the TRMs, if you were keeping track. They're part of the adaptive immunity in human tissues. They protect against reinfection with pathogens, but they're also oftentimes the basis of some autoinflammatory diseases like psoriasis, for one. And the skin, because it's all over, it's one of the biggest reservoirs of these TRMs. They're in many organs, but they're in the skin everywhere, of course. and they control skin resident memory. And that's critical for regulating normal vigilance, but also for treating these chronic inflammatory conditions, like I said, psoriasis. So there's been a lot of study of them. You look at them in the mouse organs, and you can show that there's these tissue-resident cells. But of course, like we're talking about mice, and it's an issue because mice, they don't live forever, or they're their preferred model, though. So we don't really know about the real long-term biology of these cells. Although it's suggested because we have decades-long immunity to these common pathogens, you would think that these T cells, the TRMs, either they persist or they self-renew in human tissues for a long time. So in order to address this, because the mouse model was falling short, and of course you can't do it in vitro, Jörg Stary, I don't know if that's how I pronounce his name. I'm going for it because he's from the Medical University of Vienna. So I think that's how they do it in Vienna, Jörg. Anyway, his group, they used allogeneic hematopoietic stem cell transplantation to ask the question, okay, this is innovation. You can't do it in a mouse, so let's do it in humans, but in a way that's totally ethical. Okay, we're going to talk about ethics on this show. What they did is they looked at these transplants because the host and donor-derived immune cell populations can be tracked separately. You know which is which, of course. And furthermore, looking in these patients, you can get the secondary measures and of the role of these TRMs in graft-versus-host disease, okay? And that's really the clinical endpoint here that I think is really exciting, and we're going to come back around to that. Anyway, what they found was that effectively they did some single-cell-seq. Of course, got to do it. If you're going to cover a story on the show, it has to have some high-tech. Arun, take a lesson. I'm just kidding. But they did this in-depth analysis of the blood and skin samples from a large cohort of patients before and after allogeneic hematopoietic stem cell transplantation, showed that these T cells persist for more than 10 years, all right, 10 years. Whereas the ones in the peripheral blood, they are like very high turnover. And they kind of did all this single-cell-seq to show that there was a unique survival program. There were these unique markers so that we can kind of prognostically or prospectively isolate them in future studies. So that's cool. And they showed also just, you know, meat and potatoes that there's clones of them. They expand over time. And the real thing that's amazing here is that they implicated a role for these cells in local inflammation. And not just the local inflammation, but in local inflammation that underlies graft-versus-host disease, okay? And this is a big deal because the dogma persists that it's graft-versus-host disease in many cases, most cases, right? Which means that it's a transplant of the hematopoietic cells that then populate the skin and start chewing stuff up, right? But the reality here suggests that it's the residual TRM cells that can release from the host, that can release inflammatory molecules that then trigger a lot of inflammatory processes that are related to GVHD. So it's a nice, I like experimenting in humans type of thing. I love that when it's done right. And then an insight here, you know, just basic, like the tools now to study this in the future, and also the real, I think, big picture insight of that maybe we got to think the semantics of graft-versus-host disease.
Daylon loves experimenting in humans when it's done right. I think we'll have to talk to Chris Scott about that one, Daylon. But yeah, hey, look, I'm not an immunologist, okay? I'll preface everything I'm about to say by saying that. But I can still see the beauty of this study for a couple reasons. One, it shows the differences between human and mouse. Like, we're using all these, you know, mouse model systems to study these issues when it comes to immunology and these tissue resident memory T cells, but there really is a value for doing the human studies, right? And the other thing that was really exciting to me was that the fact that these T cell clones can actually stay stable and functionally competent for 10 years. That's amazing.
Yeah. 10 years is a long time. You know what's longer? I'll tell you, it's parenthood, buddy. Now you know, sucker. And that is human experimentation as far as I'm concerned. So now you're as unethical as I am, all right? Tell Keon about experimentation with his life. All right, let me stop. So I'm going to give you guys a message from Stem Cells quickly before we get to our interview with Chris Scott. We can talk about whether or not we're experimenting in the right way. Do you study mesenchymal stromal cells, also known as MSCs? There are currently over 350 active clinical trials utilizing MSCs, but these cells have long been a source of confusion and exploitation. Yeah, we're going to talk about that with Dr. Scott. Leaders in the field are now calling for better methods for MSC identification and characterization. To learn more, read about their latest commentaries and summaries of key MSC publications at www.stemcell.com/MSC updates. All right, guys, today we have with us Dr. Christopher Scott, who is the Dalton Tomlin Professor of Medical Ethics and Health Policy at Baylor College of Medicine. Dr. Scott uses qualitative and quantitative approaches to study the ethical, legal, social, and policy implications of emerging biotechnologies and clinical medicine. Christopher, thank you so much for joining us.
Thank you for having me, Dalen and Aaron.
Yeah, Chris. Well, we'll jump right into it. You're one of the stem cell field's leading bioethicists, even though you've kind of branched out as of late. And I've brought it up a lot, actually. A big reason why I love stem cell biology is because of the great discussions that we can have about different technologies, right? There's so many cool technologies that have emerged over the last decade. And we've actually written a couple of articles together about genome editing of human embryos and also brain organoids recently. So we'll start off with a broad question. As a bioethicist, what stem cell related or unrelated technology excites you the most right now, since it gives you the most to think about and talk about as an ethicist?
Well, the thing that gets me up in the morning really is the advances in human genome editing, and it has a strong kind of stem cell connection. So it brings two technologies pretty close together. And for me, that's like kid in the candy store, you know. The fact that we now have a couple of Nobelists to thank this year makes it really all the sweeter. And human genome editing is one of those technologies from an ethics and policy point of view that is multidimensional. It crosses over and through boundaries, countries, labs. It's one of these so-called democratizing technologies, as were some types of stem cells, where many people use them, very widespread. And so when you get that kind of diffusion of technologies like we saw with stem cells, you get some really interesting social and ethical phenomenons. One of the things that, Aaron, you and I talked about a lot was the so-called phenomenon of stem cell tourism. You remember that? Where patients who could not find a therapy for whatever reason, because they were closed out of a clinical trial or because it hadn't been approved by their government, would travel to another jurisdiction to get that therapy. And it might be in a place where regulations were really permissive or non-existent. And we just had a really big meeting with 35 of the world's leading genome editing people. Many of them were very active in the stem cell debates of 10 years ago. And one of the questions we asked them in this meeting was, what's on the near horizon that worries you the most? And these are stem cell scientists and lawyers and ethicists and social scientists, religious leaders. And one of the responses that kind of rose to the top of all of the discussions we had was, this might be the stem cell tourism on steroids that emerged in 2005 and is still going on today. And on steroids, we mean that this CRISPR technology is so widely spread. I mean, everybody is using it. And it doesn't take a 20-year trained expert to familiarize themselves with CRISPR technology. A lot of people can do it. And so because it's so diffuse, and so many diverse people are using it, the experts that we talk to think that we may have unregulated CRISPR clinics hanging out, where you could go engineer yourself, or perhaps in the most extreme case, perhaps engineer an embryo and the resulting baby, like He Jiankui did in China a couple years ago.
Yeah, I want to jump in there because, it must be exciting. I'll say it's exciting to be, as you said, it's like a kid in the candy store as these technologies converge. But it's also kind of harrowing, I bet, to be the one people look to, to address these kind of questions, because you're supposed to have the final say. And specifically in your case, it's the pace of advance, I would imagine, that could make your head spin. It was 5 years ago you and Arun wrote that piece on modification of the beta globin locus, right? And that 5 years in science time is like an eye blink, right? And that was discarded triploid embryos with a success rate 4 correctly targeted out of 28, which is essentially like, it's feasible, but it doesn't really work that well.
Yeah.
And a few short years later, designer baby headline that's been floating around in people's imagination is a hard reality. Is it a challenge keeping up, not just with like the tech and the pace of the tech, but the implications of that tech? Do you find that you're kind of like, on your heels when you think and talk about these things a little bit?
Absolutely. I'll tell you a story of a colleague that Arun and I have at Stanford University, Vittorio Sebastiano, stem cell biologist, also a CRISPR scientist. So, there are these hints that come on to the horizon that something's going to happen. And one of those hints was the paper that Arun and I talked about, those triploid embryos. But there were hints before then, right? So in 2014, you guys may remember this cover article of Cell. And on the cover was this adorable pair of twin monkeys.
Yeah.
What were they, cynomologous monkeys?
Right. Yeah, I think so.
And the cover article was a group from China who had successfully engineered, using CRISPR, a couple of primates. And scientists, you guys know, will tell you something about the species barrier as kind of that last leap before you get into human applications. And that was that leap from mammals to primates, that paper. And I was sitting in my office. I don't remember what I was doing, looking out the window probably, but Vittorio came in and he slapped that Cell paper down on my desk and said, "This is going to happen in humans, and it's going to happen in China." He said that in 2014, so fully 5 years before, or almost 5 years before we heard of the controversy in China. So the question that policy scholars and ethicists ask with this is, we saw it coming, or we saw hints of it coming. Why couldn't something have been done to prevent or discourage or whatever this sorts of behavior? Now, the rejoinder to that is, you're never going to kind of stop everybody or the lone rogue actor from doing this. But my counter argument is that you could put some things in place that would discourage something like that from happening, or at least give that individual, He Jiankui, some knowledge that, if I go down this road, it's going to be really bad for me. I might go to jail, or I might never practice biophysics again. He's a biophysicist. So that actually happens to be the subject of an NIH grant that we were just awarded, and we're kind of just finishing our first aim, something called anticipatory governance. How do you look far enough down the road to identify those things that are plausible, not crazy, but just plausible? And then how do you prepare yourself for the eventuality that those sorts of things may happen, knowing full well that many of them won't? And with all that kind of that armamentarium of things that could happen, you can actually start to do responsible governing before it happens, and be ready when something like that comes out of the gate. So
yeah,
it's a good question.
It's about the regulation, right? So that kind of what it comes down to. And it's funny that you mentioned Vittorio, coming into your office and throwing that paper down on your desk in 2014. And, here we are 5 to 6 years later, and he was right, and we were right. And that's exactly what happened. Human edited human embryos and CRISPR'd human children were born in China, right? And this is something that happened within the last few years. And at least in my opinion, and maybe it's because we're living in this crazy current reality of 2020, I feel like it's faded to the background a little bit. And that's a little mind-blowing to me, because I think this is like a species-altering event that happened within the last 2 to 3 years. We should be paying more attention to it, right? But what you talked about the regulation. What can we do, like as a society, so that we can better understand some of this technology that's emerging and the importance of regulating it? Yeah. And then what can we do as a government?
Well, I'll just say it's not always about just regulation. We tend to kind of think that there's going to be a law, and then if a law happens, or if a guideline happens, or if we have some regulations that this is going to go away or be helped in some way. Well, I think it's bigger than that. I think it's also about behavior. It's about values. It's about who ought to be making these decisions, how we engage other individuals besides just scientists and ethicists in these questions about how we govern these technologies. So to the question about how we would do these sorts of things, one thing I think that can help is that we deliberate about this stuff early and often. So you can't just do it once and then walk away and think, oh, we've had our discussion. And that's a model that science policy has had for decades. National academies of science, they convene their big boards of experts. They take 6 months to get everybody together, and then they meet for 2 days, and then they issue these monumental reports, and some of them are very good. But that is a really slow process, right? And those individuals that populate those groups, I know many of them, and I've been to National Academies meetings. They're the usual suspects, right? So you get this kind of monoculture of experts talking about something that affects everybody. And my view, and I think the increasing views of many social scientists and ethicists, are that this needs to be more deliberative. It needs to be more democratic. And it needs to include the concerns and values of individuals who stand to use and perhaps benefit from these. And that's an easy statement to make and really, really, really hard to do.
Right. Yeah, I mean, that's the challenge, right? There's all these bodies that you can convene, and there's regulations that you can encode, and you can have oversight, and you can have whatever punitive measures. But at the end of the day, it's the people, right? It's the people doing the work that are, the problem and the solution. I remember Huang Wusuk cloning fiasco that dominated the science headlines a couple decades ago. And then there's been a series of these heartbreaking frauds along the way, like, for example, the STAP controversy and subsequent suicide by an icon, a great scientist who killed himself because of this controversy, Yoshiki Sasai. And many of these debacles seem to have begun with breakdowns in your garden variety research ethics. And I think a part of that is because the stakes are so high. It's a really competitive environment, and the upside is momentous, right? So you think that the ethical fabric, if you had to compare the ethics of any researcher to another, do you think that like the ethical fabric is strained on an individual or maybe even a collective level? Here right now in the midst of this pandemic, we need the vaccine to work, right? Do you think there's a strain on our individual or collective ethics when the upside is so necessary or momentous? Like, in other words, do you think that there's more research misconduct in these high visibility ventures?
Yeah, that's a good question. I don't know. I think when you have a high reward situation, like the Wang scandal, and have an individual that is suitably motivated to cheat, to get to that, whatever it is, international fame, certainly for stem cells. I think for He Jiankui, it was the same kind of sort of thing. He wanted to be the first. That's how he talked about it. That is a crucible for an event like the ones we've talked about. Whether that makes collectively scientists kind of more at risk about this, I don't know. I mean, it's a great research study to actually ask that question. I'm an optimist mostly, even though I'm in ethics, and most scientists are really, really good people. I'm a former scientist. I'm a former bench researcher, and some of the most fabulous, thoughtful individuals I've ever met are in the sciences, bench sciences. So I think everybody wants to do the right thing. David Baltimore said an interesting thing at the Hong Kong summit where He Jiankui announced his results, and I think it was when he wound up the meeting, he said, this has been, I'm paraphrasing, this has been a failure of the scientific community to police itself or something like that. So he was like turning around, looking right at the audience and saying, it's the scientists are responsible for this, not anybody else. It's our house. Yeah. We have to do something about this. So there's some, there's a self-own and a collective own with a statement like that. But as I've learned from this project with the NIH, some scientists don't want to go there. They said that's your job, Chris. It's your job to tell me what's right and wrong, what to do. And it's like, oh, great, okay. Others say, for example, an editor of a very good journal told me, absolutely, we do not want scientists to govern themselves. They're horrible at it. And you can have them weigh in because they know the technology and the rest, but don't have them govern them. Don't have them police themselves because they can't do it. So.
Yeah, I mean, I can attest to that myself. I'm a bit guilty with my, IACUC protocols and whatnot. I can always come up with a good reason why the work is essential and the pain and suffering of the animal is minimized to the absolute, minimum. But yeah, like at the end of the day, I shouldn't be making that call, right? Because I want to do that experiment.
I think a lot of it has to come down to the training, right? I think you have to be able to emphasize during the training process as flawed as it is during grad school, you have to emphasize that this stuff is important. And especially if we're working in a field as cutting edge as pluripotent stem cells or genome editing, we have to teach the kids, we have to teach the students that you need to think about this. Again, it's part of the reason I love this field, is that we were able to have these discussions, but they have real world impacts, as we're seeing right now, right? And so speaking of like high visibility, high reward science, iPSC has really turned the stem cell field upside down, right? I guess it's been about 15 years. And for good reason. You can turn pretty much anything into anything else. You can pretty much turn any somatic cell into a pluripotent stem cell. So game-changing, Nobel Prizes got awarded. But there's actually one unique application of iPSCs that I don't think has gotten too much attention as of late, and perhaps that's because of the difficulty of the approach. And that's actually the production of gametes, sperm and egg, from iPSCs. I actually remember writing a paper on this topic when I was in your ethics class, and it was really cool to think about. And Dalen's out there in the Cornell Center for Reproductive Medicine, so I thought he'd get a kick out of the question as well. And so what would it mean? What would it mean to be able to generate functional human gametes from a person's own skin or blood, like derived iPSCs, and how might it end up changing, I don't know, our species as a whole?
Well, you've hit on two really interesting things, and you're exactly right. The generation of gametes is one of those corner areas of both science and ethics that there's been some discussion about it, but not a lot written. I think it's fascinating. iPSCs, thanks to Shinya Yamanaka, have revolutionized the way that we think about fate, cell fate, and how we organize tissues. And the fact that you could drive a cell backwards and then forwards into a gamete, I think is just a stunning sort of thing to contemplate. Let's talk about it from the developmental point of view, right? So many diseases of early development that could be studied through this sort of mechanism. So don't even think about kind of the end product where you could actually maybe generate a baby using the kind of the twin pathways of gamete production, but just understanding failures of early human development in IVF, I mean, Dalen, for sure, would be super important sorts of things to investigate. But when you have that sort of power, you have a similar sort of thing that you have with human genome engineering, in my view, and that's you can twiddle with that process any number of ways, changing genes, putting in genes, cutting out genes, and so you're doing kind of a high level heritable, and that's kind of the golden word, gene engineering with gametes. And these sorts of things will be with the end product, whether it's a baby, or that baby when he or she grows up, and the progeny of that baby forever. So that's a real, I think, one of the things that ethics takes a deep breath about, right, are the heritable parts of these technologies that we're looking at.
Yeah, right. Arun alluded to it earlier. It's like a new species. So yes, on that point regarding the IVF, we had Katsuhiko Hayashi at our center, who is famously the lead author on a bunch of the Mitsunori Saito work, generating gametes, and then he started his own lab, and he was at our center right after he had made human iPS cells into oocytes, using like mouse fetal gonad. It was amazing. And for everyone in the group there, which was really clinicians, they were like, okay, so what's next? Like, when are we going to see the first, because, clinicians, I mean, I wish I were a clinician sometimes. They got all the money. But anyway, I digress. It was amazing. And needless to say, the implications, as you alluded to, mind-bending. We talked about in the meeting about the idea of getting an XY genotype background, but you could create eggs from that background because it's the humoral factors that would direct the formation of the gametes. The idea there being that like two gay men could share biological offspring, one contributing the sperm and one contributing skin that becomes egg. It's like truly mind-bending. If the implications though are huge, of course. But the real thing there that blew my mind was that Katsuhiko, he told me that there was no funding for his work. And I'm paraphrasing him here, but he said the bottom line was that those opposed generally to the idea for good reason because of heritable germline manipulation. They found the idea repellent, and I can understand that. But the other thing that was more surprising to me, he said that in his society, in Japan, in general, the discussion of reproductive challenges was not discussed openly. And there's there's a real kind of a repressive atmosphere surrounding IVF or assisted reproduction, and thus research funding toward that end. And he's doing fine just now. I just saw his group was recommended for $2.5 million from Open Philanthropy. But that kind of tells a story. He had to go in one of these more private philanthropic apparatus. And the question here I'm talking about is like, I was surprised because that culture versus in the U.S., I don't think it would be. I mean, I know it's not, we're very open about IVF and assisted reproduction here. Do you find that the ethics vary from culture to culture? Like you're having the same kind of discussion about the same thing, but the nature of the discussion is very different depending on where you're having that discussion. How do you reconcile that with this whole idea of the global scientific community? I mean, we have to reach consensus, but we have really disparate views when it comes to our culture. You know what I mean?
Yeah, that's a great question. In ethics, we call this kind of the normative view. What's the norm for where you are? And our norms are different than Japan's, which are different than China's, which are different than Islamic countries, which are different than Catholic countries. And as research becomes more global and interconnected and more democratic, you are faced with this thicket of problems that face the scientists who are trying to work together across these kind of gradients of ethics and regulation, and for the patients who might eventually use them. So just like with COVID-19, Americans are going to get an American vaccine, looks like. And with stem cell therapies, you will find, and we did find, that the benefits of some of those therapies were derived by the people in the jurisdictions where they were invented. And in fact, some of the research that I did early on with stem cell lines showed that the lines where they were derived, they tended to stay put there. So there was a strong kind of geographic anchor for certain stem cell lines to stay in certain countries. And that can be a good thing and a bad thing. But for just plain things like collaboration, there were times during the stem cell debates where researchers weren't sure whether they were liable for criminal charges by collaborating with someone in another country where embryonic stem cell research was banned.
Wow.
It wasn't clear by the law. Sometimes the law was silent, and sometimes there was, stuff that a able lawyer or a scholar of law could say, yeah, I wouldn't do that if I were you. Mm-hmm. Just because some attorney general somewhere might get, a bee in his or her bonnet and say, here's the law.
My gosh, they're gonna lock up George Daly any day now.
Good luck. Oh man. So science is definitely worldwide, right? As we've talked about, Chris. And I want to come back to your optimism for a second here. You've said that scientists in general are good people trying to do good things, right? And unfortunately, it seems like we have to discuss science with the bioethicists when there's the potential for something bad to happen with a new technology or a new discovery, like when there's the potential to misuse a new technology. But as an ethicist, you have to appreciate the scientists who have discussions with you throughout the development process of a technology. So can you think of some examples where that was a situation and the scientists got it right and they were really working with the ethicists from the start when it came to actually these potentially groundbreaking or controversial new technologies?
Yeah, a good stem cell case jumps to mind. And this is someone that you guys both know, Irv Weissman. So Irv was contemplating an experiment that would put human, let's see, I believe these were oligodendrocytes that were derived from human embryonic stem cells into a mouse fetus.
Yeah, I think that's right. Yeah.
Right? Mm-hmm. And I think he even was contemplating an earlier developmental stage, not an embryo, but something kind of between. And before he did the experiment, he knew the cultural and perhaps religious sensitivities around an experiment like this, because he was, in essence, creating a chimera, human-animal chimera, and he was using perhaps the most ethically fraught cell other than gametes in the body, a neuron, right? Brains, consciousness, personhood, all the stuff that kind of gives us pause, creeps us out maybe a little bit. And so before he did the experiment, he came to a group at Stanford that sits virtually in the medical center. It's called the Bedside Consulting, Bedside Ethics Consulting Group. And I was a member of that. A couple of colleagues of mine, a rather famous and good friend, Hank Greeley, is on that group. And Irv, I think the story goes, Irv went to Hank and said, since they're neighbors, and they were talking over the fence, and he said, could you put a group of people together to think about this from the ethics, law, and social vantage point, this experiment, before I do it? And he explained the experiment, and I think shared a protocol with Hank. And Hank took it and formed a big group of people at Stanford. And over the course of a few weeks, they kind of deliberated on it. They talked about as you say, Aaron, some of the more worrisome parts of this, imputing some sort of human, perhaps, consciousness into a mouse, and how you would measure that. How would you know, right? A famous veterinarian told me once that she had looked into the beady eyes of millions and millions of mice, and she has no idea what they're thinking, even in the best of circumstances. And they thought long and hard about it, and these are all in a series of great papers, by the way, that you can read about their process. And long story short, they did not think that the way that the experiment was designed would have given that mouse human consciousness for a number of reasons. And those reasons were grounded pretty strongly in developmental biology. Whisker barrel genes are going to drive that neuron to a whisker barrel, not to, an appreciation of Mozart. And the architecture of the mouse, that developmental program, was going to be so strong that it would overwhelm whatever millions of cells that would choose to put into the brain of that developing fetus. And so they gave him a green light. And the paper that Hank wrote afterwards, the title of it is Hi, I'm Mickey. And it's basically a reflection of what an ethicist, in this case a lawyer, Hank, goes through when they're picking apart something like this and asking questions about whether the experiment should proceed. And by the way, the rationale for the experiment was really good, right? I mean, if you could make mouse models of human brain disease that are really robust. Parkinson's patients make lousy research subjects. That's unethical, right?
Right.
Go experimenting on demented patients. But if you could have a real robust animal model of the human dementia, be a cool thing.
Yes. I mean, I've done a little bit of chimera work myself in my doctoral work, and I remember those conversations. And I want to preface this by saying that I recognize that the great hubris of all scientists, maybe, but myself for certain, is like the unknown, not recognizing the unknown unknowns, thinking you have it figured out. This isn't a risk, and you don't really know. So I want to preface it with that. But what I'm saying is that a lot of those conversations I left with kind of like a bit of an eye roll because of the kind of, the ideas that were being batted around, like, oh, the personhood and all that stuff. And I recognize that it's not my job to address that. I'm just going to ask for permission. You need to get some qualified people to give me the green light or not. But still, the cynical part of me was walking away like, oh, please. I'll say that. But, what I'm getting at there is that for me, at least, there's these kind of things that are kind of like outside possibilities that are kind of like theoretical conversations. Like, oh, what if you put the stem cell in the brain, but it migrated to the gonad and became sperm, and another animal had an egg, and then the two mice they got in the same cage. I mean, it's like one thing after that and the other, and you're like, oh, please. But then there's the other side where it's like, no way. That's a clear breach of ethics. And then there's like in the middle, in the gray area. So just for my curiosity and that of our listeners, as someone who's kind of seen it all, what are like what's at least one hard boundary in the ethical conduct of science and application of discovery, whatever you want to call it? What's a hard boundary for you? And what, the converse, what are some of like the gray areas that you agonize over when you're throwing one back with your colleagues? Like, I mean, I just really don't know. Is this really ethical? Could you give me a couple examples of those?
Yeah, well, ethics is everything in that gray category. That's why I like it so much. This is not a place where there are a lot of bright lines. And I've been on the ethics side of science for going on 20 years or so. And I love it so much because you do have these kind of competing things that you're trying to make a decision about. And sometimes there are no good answers. And in fact, the class that Aaron took with me, those were cases, as I recall, where I purposely selected them because there's no answer. You have to kind of come up with your own rationale, and you can argue it really well from both sides. But for a bright line, I guess I would say, He Jiankui did a bad thing. I mean, this is a bad thing. You don't go making babies when you don't have the risks sorted out. And it was a basketful of misdeeds. I mean, the ethics books are going to be written about this forever. He forged a signature on an ethics review. The informed consent lied about what the experiment was. He called it a vaccination. So, the germline editing things for me is a hard stop, not a permanent hard stop necessarily, but right now, that's a no-go zone. Stem cells, I have to think about that. I think when the risks haven't been properly evaluated, especially with embryonic stem cells, I think there are probably some human experiments that simply shouldn't take place. Same sort of kind of rationale. You don't want to be putting a potentially cancerous cell in an unregulated environment and creating a tumor someplace. And an interesting kind of twist, I think that's how iPSCs kind of gained the traction.
Right.
Yamanaka, I think at one point said that it's because this embryonic stem cell environment is so morally fraught for him and for society that he wanted to pursue a different way. So.
Right. Yeah, but I mean, I think something we talked about earlier, certainly I think there are certain hard lines that we don't want to cross, but because we are an international community now, right, there are cultural differences between where those lines should be drawn. So I think it's hard to get a consensus, is what I'm saying. It's very difficult to get a consensus across the field, but we're trying, and you're trying, and we're very glad that you are trying, because these are important questions that we should have, and it's why it's so exciting to be involved in this field as a trainee and as a veteran scientist. So, well, Chris,
I really appreciate that comment a lot. I think it's by talking to scientists where I kind of gain a lot of my ethical frameworks. Right. You can sit off to the side and be a philosopher and think about all of this kind of in the abstract, but it doesn't do you much good, I think, when it comes down to kind of making real world decisions or giving advice.
Right. It comes down to the science and what happens in the science. So, Chris, thank you so much for joining us here today. It's been a pleasure to catch up with an old mentor and an old friend of mine from my grad school days. And it's you're the first person I go to whenever I have a question or something that I'm excited about when it comes to a new technology that's either in the stem cell field or outside of it. So I'm really happy that you're able to join us here today.
Thank you very much, Aaron. Thanks for having me. And Dalen, nice talking to you too.
Likewise. All right, you guys, that brings us to the end of this show. A little something different today. I love talking to someone about ethics, though, an expert on ethics, because the conversation could go on forever. It went on a bit long, but I think it was a lot of fun. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at STEMCELL or by email at info@stemcellpodcast.com with feedback or to suggest guests. You guys get out there and do your work, but keep it ethical, please. And tune in a couple weeks for our next episode. Thanks for listening.